Unit 7 System Physiology Animal. A. Blood and circulation, blood corpuscles, hemopoiesis and formed elements, plasma function, blood volume regulation, blood groups, hemoglobin, immunity, hemostasis. Components of blood. Blood is a highly differentiated, complex living tissue that pulsates through the arteries to every part of the body, interacts with individual cells via an extensive capillary network, and returns to the heart through the venous system. Many of the functions of blood are undertaken in the capillaries, where the blood flow flows dramatically, allowing the efficient diffusion and transport of oxygen, glucose, and other molecules across the monolayer of endothelial cells that form the thin capillary walls. In addition to transport, blood in the cells within it mediate other essential aspects of immunity and hemostasis. The human body is continually invaded by pathogenic microorganisms that enter through skin cuts, mucus, membranes, and other sites of infection and tissue. Disruption. To oppose pathogenic microbes, the body has developed a highly sophisticated immune system. Cells of the immune system, the white blood cells, are derived from bone marrow precursors and are delivered to their sites of action by the blood. These cells, also known as leukocytes, exert their effects in conjunction with antibodies and protein cofactors in blood. In this chapter, we will see how certain leukocytes act without prior sensitization to neutralize offending pathogens, while others require a prior infectious insult to deal with invaders. In addition to infectious assault, the body is continually threatened by the devastating consequences of vascular leak or hemorrhage as a result of even the most innocuous tissue injury. A highly organized clotting system, consisting of blood platelets that work in conjunction with blood plasma clotting factors, prevents excessive fluid loss by rapidly forming a hemostatic plug. In addition to physically constraining fluids within ruptured vessels, platelets release potent biological cofactors during the development of this hemostatic plug, which promote wound healing, prevent further infection, and promote the development and vascularization of new tissue. The components of blood. Blood is an opaque, red liquid consisting of several types of cells suspended in a complex, amber fluid known as plasma. When blood is allowed to clot or coagulate, the suspended medium is referred to as serum. Blood has a higher density and viscosity than water. Blood is normally confined to the circulation, including the heart and the pulmonary and systemic blood vessels. Blood accounts for 6 to 8% of the body weight of a healthy adult. The blood volume is normally 5.0 to 6.0 L in men and 4.5 to 5.5 L in women. The density or specific gravity of blood is approximately 1.050 grams per milliliter. Density depends on the number of blood cells present and the composition of the plasma. The density of individual blood cells varies according to cell type and ranges from 1.115 grams per milliliter for erythrocytes to 1.070 grams per milliliter for certain leukocytes. While blood is only slightly heavier than water, it is certainly much thicker. The viscosity of blood, a measure of resistance to flow, is 3.5 to 5.5 times that of water. Blood's viscosity increases as the total number of cells present increases and when the concentration of large molecules, macromolecules, in plasma increases. At pathologically high viscosity, blood flows poorly to the extremities and internal organs. Functions of blood. While the cellular and plasma components of blood may act alone, they often work in concert to perform their functions. Working together, blood cells and plasma proteins play several important roles, including transport of substances from one area of the body to another. Immunity, the body's defense against disease * hemostasis, the arrest of bleeding. Homeostasis, the maintenance of a stable internal environment. Transport. Blood carries several important substances from one area of the body to another, including oxygen, carbon dioxide, antibodies, acids and bases, ions, vitamins, cofactors, hormones, nutrients, lipids, gases, pigments, minerals, and water. Transport is one of the primary and most important functions of blood, and blood is the primary means of long-distance transport in the body. Substances can be transported free in plasma, bound to plasma proteins, or within blood cells. Oxygen and carbon dioxide are two of the more important molecules transported by blood. Oxygen is taken up by the red cells as they pass through capillaries in the lung. In tissue capillaries, red cells release oxygen, which is then used by respiring tissue cells. These cells produce carbon dioxide and other wastes. The blood also transports heat. By doing so, it maintains the proper temperature in different organs and tissues, and in the body as a whole. Immunity. Blood leukocytes are involved in the body's battle against infection by microorganisms. While the skin and mucous membranes physically restrict the entry of infectious agents, microbes constantly penetrate these barriers and continuously threaten internal infection. Blood leukocytes, working in conjunction with plasma proteins, continuously patrol for microbial pathogens in the tissues and in the blood. In most cases, penetrating microbes are efficiently eliminated by the sophisticated and elaborate antimicrobial systems of the blood. Institute for Advanced Studies, Job 1. Unit 7 System Physiology Animal. Hemostasis. Bleeding is controlled by the process of hemostasis. Complex and efficient hemostatic mechanisms have evolved to stop hemorrhage after injury, and their failure can quickly lead to fatal blood loss, exsanguination. Both physical and cellular mechanisms still participate in tilde hemostasis. These mechanisms, like those of the immune system, are complex, interrelated, and essential for survival. Homeostasis. Homeostasis is a steady state that provides an optimal internal environment for cell function. By maintaining pH, one-on concentrations, osmolality, temperature, nutrient supply, and vascular integrity, the blood system plays a crucial role in preserving homeostasis. Homeostasis is the result of normal functioning of the blood's transport, immune, and hemostatic systems. Functions of plasma, which contains many important solutes. Plasma is composed mostly of water, 93%, with various dissolved solutes, including proteins, lipids, fats, carbohydrates, amino acids, vitamins, minerals, hormones, wastes, cofactors, gases, and electrolytes. Table 1. The solutes in plasma play crucial roles in homeostasis, such as maintaining normal plasma pH and osmolality. Some components of plasma. Normal. Concentration. Class substance range. Cation sodium, not 136 to 145 mec. L potassium K, 3.5 minus 5.0 mec. L calcium Ca asterisk, 4.2 minus 5.2 mec. L magnesium Mg asterisk, 1.5 minus 2.0 mec. L. Iron Fe 50 to 170 p. G per deciliter copper Cu. Trademark 70 to 155 pg per deciliter hydrogen H, 3545 mol L. Anions chloride C7 95 to 105 meg. L bicarbonate HCO 73 22 to 26 meg. L lactate 0.67 1.8 meg. L. Sulfate 5024 underscore 0.91.1 meg. L phosphate 3.0 4.5 milligrams per deciliter. HPOYH, PO3. Proteins total 6 to 8 grams per deciliter. Albumin 3.5 minus 5.5 grams per deciliter. Globulin 2.3 minus 35 grams per deciliter. Fat cholesterol 150 to 200 milligrams per deciliter. Phospholipids 150 to 220 milligrams per deciliter. Triglycerides 35 to 160 milligrams per deciliter. Carbohydrates glucose 70 to 110 milligrams per deciliter. Vitamins. Vitamin B greater than 200 to 800 pg per milliliter. Cofactors and vitamin A 0.15 minus 0.6 pg per milliliter. Enzymes vitamin C 0.4 minus 1.5 milligrams per deciliter. 2. 3-diphosphoglycerate 3 to 4 mmol. DPG. Transaminase SGOT 940 W per milliliter. Alkaline phosphatase 20 to 70 Li. L acid phosphatase 0.5 2 WL. Other substances creatinine 0.6 minus 1.2 milligrams per deciliter. Uric acid 0.18 minus 0.49 mmol. L blood urea nitrogen 7 to 18 milligrams per deciliter. Lodine 3.5 minus 8.0 pg per deciliter. Company 23 to 30 mmol. L bilirubin total 0.11.0 milligrams per deciliter. Aldosterone 3 to 10 nanogram per deciliter. Cortisol 5 to 18 pg per deciliter. Ketone 0.2 minus 2.0 milligrams per deciliter. There are 3 types of blood cells. Blood cells include erythrocytes, red blood cells, leukocytes, white blood cells, and platelets. Thrombocytes. Each microliter, a millionth of a liter, of blood contains 4 to 6 million erythrocytes, 4,500 to 10,000 leukocytes, and 150,000 to 400,000 platelets. There are several subtypes of leukocytes, defined by morphological differences. Figure 1, each with vastly different functional characteristics and capabilities. Table 2 lists the normal circulating levels of different blood cell types. Of the total leukocytes, 40 to 75% are neutrophilic, polymorphonuclear multinucleated cells, otherwise known as neutrophils. These phagocytic cells actively ingest and destroy invading microorganisms. Eosinophils and basophils are polymorphonuclear cells that are present in low numbers in blood, 1 to 6% of total leukocytes, and participate in allergic hypersensitivity reactions. Mononuclear cells, including monocytes and lymphocytes, comprise 20 to 50% of the total leukocytes. These cells generate antibodies and mount cellular immune reactions against invading agents. EE2. Eosinophil basophil. underscore JJ. Granulocytes. RN. LGL large granular. Lymphocyte null cell NK cell mature helper inducer suppressor. PA natural killer cell B cell T cell underscore. Lymphocytes. Monocyte macrophage. M types of leukocytes in blood and tissues. All of the cells shown here are found in the circulation except the macrophage, which differentiates from activated monocytes in tissue. Circulating blood cell levels. Blood cell type approximate normal range erythrocytes cells L. Men 4.3 minus 5.9 by 10 degrees. Women 3.5 minus 5.5 by 10 degrees. Leukocytes cells per milliliter 4,500 11,000 neutrophils 4,000 7,000 lymphocytes 2,500 5,000 monocytes 100 1,000 eosinophils 0 to 500 basophils 0 to 100 platelets cells place 150,000 400,000. Institute for Advanced Studies Jodhpur. Unit 7 System Physiology Animal. The number and relative proportion of the leukocyte subtypes can vary widely in different disease states. For example, the absolute neutrophil count often increases during infection, presumably in response to the infection. Eosinophil counts increase when allergic individuals are exposed to allergens. Lymphocyte counts decrease in AIDS and during some other viral infections. For this reason, in addition to a blood cell count, a differential analysis of leukocyte subtypes performed by microscopic examination of stained slides can provide important clues to the diagnosis of disease. Erythrocytes carry oxygen to tissues. Erythrocytes are the most numerous cells in blood. These biconcave discs lack a nucleus and have a diameter of about 7 and a maximum thickness of 2.5 The shape of the erythrocyte optimizes its surface area, increasing the efficiency of gas exchange. The erythrocyte maintains its shape by virtue of its complex membrane skeleton, which consists of an insoluble mesh of fibrous proteins attached to the inside of the plasma membrane. This structural arrangement allows the erythrocyte great flexibility as the cell twists and turns through small, curved vessels. In addition to structural proteins of the membrane, several functional proteins are found in the cytoplasm of erythrocytes. These include hemoglobin, the major oxygen-carrying protein, antioxidant enzymes, and glycolytic systems to provide cellular energy (ATP). The plasma membrane possesses ion pumps that maintain a high level of intracellular potassium and a low level of intracellular calcium and sodium. Hemoglobin, the red, oxygen-transporting protein of erythrocytes, consists of a globin or protein portion in 14 groups, the iron-carrying portion. The molecular weight of hemoglobin is about 64,500. This complex protein possesses 4 polypeptide chains, 2 globin molecules of 141 amino acids each and 2 molecules of another type of globin chain F, Y, 0, each containing 146 amino acid residues. Figure 2. Figure structure of hemoglobin. A batch molecule of hemoglobin possesses 4 polypeptide chains, each containing iron bound to its heme group. 4 types of hemoglobin molecules can be found in human erythrocytes, embryonic, fetal, and 2 different types found in adults, HbA winking phase. Each hemoglobin molecule is designated by its polypeptide composition. For example, the most prevalent adult hemoglobin, HbA, consists of 2 O chain 2B chains. Its formula is given as O, F3. HbA, which makes up about 1.5 to 3% of total hemoglobin in an adult, has the subunit formula 0, 0. Fetal hemoglobin A is a major hemoglobin component during intrauterine life. Its levels in circulating blood cells decrease rapidly during infancy and reach a concentration of 0.5% in adults. Embryonic hemoglobin is found earlier in development. It consists of 2 A chains and 2 Euro chains A, cent. The production of a chain ceases at about the 3rd month of fetal development. The production of each type of globin chain is controlled by an individual structural gene with 5 different loci. Mutations, which can occur anywhere in these 5 loci, have resulted in the production of over 550 types of abnormal hemoglobin molecules, most of which have no known clinical significance. Mutations can arise from a single substitution within the nucleic acid of the gene coding for the globin chain, a deletion of the codons, or gene rearrangement as a result of unequal crossing over between homologous chromosomes. Sickle cell anemia, for example, results from the presence of sickle cell hemoglobin HbS, which differs from normal adult hemoglobin because of the substitution of a single amino acid in each of the 2B chains. Oxyhemoglobin HbO, the oxygen-saturated form of hemoglobin, transports oxygen from the lungs to tissues, where the oxygen is released. When oxygen is released, HbO becomes reduced hemoglobin Hb. While oxygen-saturated hemoglobin is bright red, reduced hemoglobin is bluish red, accounting for the difference in the color of blood in arteries and veins. Certain chemicals readily block the oxygen-transporting function of hemoglobin. For example, carbon monoxide CO rapidly replaces oxygen in HbO, resulting in the formation of the stable compound carboxyhemoglobin HbCO. The formation of HbCO accounts for the asphyxiating properties of CO. Nitrates and certain other chemicals oxidize the iron in Hb from the ferrous to the ferric state, resulting in the formation of methemoglobin Methb. Methb contains oxygen bound tightly to ferric iron. As such, it is useless in respiration. Cyanosis, the dark blue coloration of skin associated with anoxia, becomes evident when the concentration of reduced hemoglobin exceeds 5 grams per deciliter. Cyanosis may be rapidly reversed by oxygen if the condition is caused only by a diminished oxygen supply. However, cyanosis caused by the intestinal absorption of nitrates or other toxins, a condition known as enterogenous cyanosis, is due to the accumulation of stabilized methemoglobin and is not rapidly reversible by the administration of oxygen alone. Red cell morphology. In addition to revealing alterations in absolute values, stained blood films may provide valuable information based on the morphological appearance of blood cells. Erythrocytes are formed from precursor blast cells in the bone marrow. Figure 3. This process, termed erythropoiesis, is regulated by erythropoietin, a hormone produced in the kidneys. Changes in red cell appearance occur in a variety of pathological conditions. Institute for Advanced Studies, Jodhpur 3. Unit 7 System Physiology Animal. Merck frontoexis, erythrocyte production in healthy adults occurs in narrow sinusoids. Driven by the hormone erythropoietin, the uncommitted stem cell differentiates along the erythrocyte lineage, forming normoblasts, also referred to as erythroblasts or burst-forming cells, reticulocytes, and, finally, mature erythrocytes, which enter the blood stream by the process of diaphoresis. Excessive variation in the size of cells is referred to as anisocytosis. Larger than normal erythrocytes are termed macrocytes. Smaller than normal erythrocytes are referred to as microcytes. Poikilocytosis is the presence of irregularly shaped erythrocytes. Burst cells are spiked erythrocytes generated by alterations in the plasma environment. Schistocytes are fragments of red cells damaged during blood flow through abnormal blood vessels or cardiac prostheses. The hemoglobin content of erythrocytes is also reflected in the staining pattern of cells undried. Orange throughout, with a very slight central pallor as a result of the cell shape. Hypochromic cells appear pale with only a ring of deeply colored hemoglobin on the periphery. Other pathological variations in red cell appearance include spherocytes, small, densely staining red cells with loss of biconcavity as a result of congenital or acquired cell membrane abnormalities, and target cells, which have a densely staining central area with a pale surrounding area. Target cells are thin but bulge in the middle, unlike normal erythrocytes. This alteration is a consequence of hemoglobinopathies, mutations in the structure of hemoglobin. Target cells are observed in liver disease and after splenectomy. Nucleated red cells are normally not seen in peripheral blood because their nuclei are lost before they move from the bone marrow into the blood. However, they appear in many blood and marrow disorders, and their presence can be of diagnostic significance. One type of nucleated red cell, the normoblast C figure 3, is seen in several types of anemias, especially when the marrow is actively responding to demand for new erythrocytes. In seriously ill patients, the appearance of normoblasts in peripheral blood is a great prognostic sign preceding death, often by several hours. Another nucleated erythrocyte, the megaloblast, is seen in peripheral blood in pernicious anemia and folic acid deficiency. Erythrocyte destruction. Red cells circulate for about 120 days after they are released from the marrow. Some of the senescent old red cells break up, hemolyze, in the bloodstream, but the majority are engulfed by macrophages in the monocyte macrophage system. The hemoglobin released on destruction of red cells is metabolically catabolized and eventually reused in the synthesis of new hemoglobin. Hemoglobin released by red cells that lyse in the circulation either binds to haptoglobin, a protein in plasma, or is broken down to globin and heme. Heme binds a second plasma carrier protein, hemopexin, which, like haptoglobin, is cleared from the circulation by macrophages in the liver. In the macrophage, released hemoglobin is first broken into globin and heme. The globin portion once catabolized by proteases into constituent amino acids that are used in protein synthesis. Heme is broken down into free iron, Fe, and biliverdin, a green substance that is further reduced to bilirubin. Iron recycling. Most of the iron needed for new hemoglobin synthesis is obtained from the heme of senescent red cells. Iron released by macrophages is transported in the ferric state in plasma bound to the iron transporting protein, transferrin. Cells that need iron, e.g., for heme synthesis, possess membrane receptors to which transferrin binds. The receptor-bound transferrin is then internalized. The iron is released, reduced intracellularly to the ferrous state, and either incorporated into heme or stored as ferritin, a complex of protein and ferrous hydroxide. Iron is also stored as ferritin by macrophages in the liver. A portion of the ferritin is catabolized to hemosiderin, an insoluble compound consisting of crystalline aggregates of ferritin. The accumulation of large amounts of hemosiderin formed during periods of massive hemolysis can result in damage to vital organs, including the heart, pancreas, and liver. The recycling of iron is quite efficient, but small amounts are continuously lost. Iron loss increases substantially during menstruation. Iron stores must be replenished by dietary uptake. The majority of iron in the diet ones derived from heme and meat, organic iron, but iron can also be provided by the absorption of inorganic iron by intestinal epithelial cells. In these cells, iron attached to heme is released and reduced to the ferrous form Fe by intracellular phlegoprotein. The reduced iron, both released from heme and absorbed as the inorganic ion, is transported through the cytoplasm bound to a transferrin-like protein. When it is released to the plasma, it is oxidized to the ferric state and bound to transferrin for use in heme synthesis. 4 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. Platelets participate in clotting. Platelets are irregularly shaped, disc-like fragments of the membrane of the precursor cell, the megakaryocyte. Megakaryocytes shed platelets in the bone marrow sinusoids. From there, the platelets are released to the blood, where they function in hemostasis. Several factors stimulate megakaryocytes to release platelets, including the hormone thrombopoietin, which is generated and released into the bloodstream when the number of circulating platelets drops. Platelets have no defined nucleus. They are 1/4 to 1/3 the size of erythrocytes. Platelets possess physiologically important proteins, stored in intracellular granules, which are secreted when the platelets are activated during coagulation. The role of platelets in blood clotting is discussed below. Leukocytes participate in host defense. Each of the 3 general types of leukocytes, myeloid, lymphoid, and monocytic, follows a separate line of development from primitive cells, see figure 1. Mature cells of the myeloid series are termed granulocytes, based on their appearance after staining with polychromatic dyes, such as Wright's stain. While monocytes and lymphocytes may also possess cytoplasmic granules, they are not clearly visualized with commonly used stains. Therefore, monocytes and lymphocytes are often referred to as the granular leukocytes. The nuclei of most mature granulocytes are divided into 2 to 5 oval lobes connected by thin strands of chromatin. This nuclear separation imparts a multinuclear appearance to granulocytes, which are, therefore, so known as polymorphonuclear leukocytes. 3 distinct types of granulocytes have been identified based on staining reactions of their cytoplasm with polychromatic dyes: neutrophils, eosinophils, and basophils. Neutrophils. Neutrophils are usually the most prevalent leukocyte in peripheral blood. These dynamic cells respond instantly to microbial invasion by detecting foreign proteins or changes in host defense network proteins. Neutrophils provide an efficient defense against pathogens that have gotten past physical barriers such as the skin. Defects in neutrophil function quickly lead to massive infection and, quite often, death. Neutrophils are amoeba-like phagocytic cells. Invading bacteria induce neutrophil chemotaxis, migration to the site of infection. Chemotaxis once initiated by the release of chemotactic factors from the bacteria or by chemotactic factor generation in the blood plasma or tissues. Chemotactic factors are generated when bacteria or their products bind to circulating antibodies, by tissue cells when infected with bacteria, and by lymphocytes and platelets after interaction with bacteria. After neutrophils migrate to the site of infection, they engulf the invading pathogen by the process of phagocytosis. Phagocytosis once facilitated when the bacteria are coated with the host defense proteins known as opsonins. A burst of metabolic events occurs in the neutrophil after phagocytosis. In the phagocytic vacuole or phagosome, the bacterium is exposed to enzymes that were originally positioned on the cell surface. Thus, phagocytosis involves invagination and then vacuolization of the segment of membrane to which a pathogen is bound. Membrane-bound enzymes, activated when the phagocytic vacuole closes, work in conjunction with enzymes secreted from intracellular granules into the phagocytic vacuole to destroy the invading pathogen efficiently. One important membrane-bound enzyme, nicotinamide adenine dinucleotide phosphate (NADPH) oxidase, produces superoxide anion O%. Superoxide is an unstable free radical that kills bacteria directly. Superoxide also participates in secondary free radical reactions to generate other potent antimicrobial agents, such as hydrogen peroxide. Superoxide generation in the phagocytic vacuole proceeds at the expense of reducing agents oxidized in the cytoplasm. The reducing agent, NADPH, is generated from glucose by the activity of the hexose monophosphate shunt. Aerobic cells generate reduced nicotinamide adenine dinucleotide (NADH) and ATP. When glucose is oxidized to carbon dioxide, the hexose monophosphate shunt operates in neutrophils and other cells when large amounts of NADPH are needed to maintain intracellular reducing activity. Oxygen reduction by the NADPH oxidase that generates superoxide in neutrophils is driven by an increased availability of NADPH after phagocytosis. NADPH plus 205 to 202 tilde plus NAD asterisk plus HT. NADPH equals X equals E. A complex set of biochemical events unfolds after phagocytosis to activate the neutrophil NADPH oxidase, which once dormant in resting cells. The oxidase is activated by its interaction with an activated G protein and cytosolic molecules that are generated during phagocytosis. The NADPH oxidase is activated in a manner that allows the enzyme to secrete the toxic free radical, superoxide, into the phagocytic vacuole while oxidizing NADPH in the cell's cytoplasm. This explosion of metabolic activity, collectively termed a respiratory burst, leads to the generation of potent, reactive agents not otherwise generated in biological systems. These agents are so reactive that they actually generate light, biological chemiluminescence, when they oxidize components in the bacterial cell wall. Other bactericidal agents and processes operate in neutrophils to ensure efficient bacterial killing. Phagocytized bacteria encounter intracellular defensins, cationic proteins that bind to and inhibit the replication of bacteria. Defensins and other antibacterial agents pour into the phagocytic vacuole after phagocytosis. Agents stored in neutrophil granules include lysozyme, a bacteriolytic enzyme, and myeloperoxidase, which reacts with hydrogen peroxide to generate potent, bacteria-killing oxidants. One of the oxidants generated by the myeloperoxidase reaction is hypochlorous acid (HOCl), the killing agent found in household bleach. Granules also contain collagenase and other proteases. Institute for Advanced Studies, Jodhpur 5. Unit 7 System Physiology Animal. Eosinophils. Eosinophils are rare in the circulation but are easily identified on stained blood films. As the name implies, the eosinophile takes on a deep eosin color during polychromatic staining. The large, refractal cytoplasmic granules of these cells stain orange, red to bright yellow. Like neutrophils, eosinophils migrate to sites where they are needed and exhibit a metabolic burst when activated. Eosinophils participate in defense against certain parasites, and they are involved in allergic reactions. The exposure of allergic individuals to an allergen often results in a transient increase in eosinophil count known as eosinophilia. Infection with parasites often results in a sustained overproduction of eosinophils. Basophils. Basophils are polymorphonuclear leukocytes with multiple pleomorphic, coarse, deep staining metachromatic granules throughout their cytoplasm. These granules contain heparin and histamine, which have anticoagulant and vasolylating properties, respectively. The release of these and other mediators by basophils increases regional blood flow, facilitating the transport of other leukocytes to areas of infection and allergic reactivity or other forms of hypersensitivity. Monocytes and lymphocytes. In contrast to granulocytes, monocytes and lymphocytes are mononuclear cells. Monocytes are phagocytic cells but lymphocytes are not. Both participate in multiple aspects of immunity. Monocytes were originally differentiated from lymphocytes based on morphological characteristics. The cytoplasm of monocytes appears pale blue or blue-gray with white stain. The cytoplasm contains multiple fine reddish blue granules. The monocyte nucleus may be shaped like a kidney bean, indented, or shaped like a horseshoe. Frequently, however, it is rounded or ovoid. Upon activation, monocytes transform into macrophages, a large, active mononuclear phagocytes. Morphologically, circulating lymphocytes have been assigned to 2 broad categories: large and small lymphocytes. In blood, small lymphocytes are more numerous than larger ones. The latter closely resemble monocytes. Small lymphocytes possess a deeply stained, coarse nucleus that is large in relation to the remainder of the cell, so that often only a small rim of cytoplasm appears around parts of the nucleus. In contrast, a broad band of cytoplasm surrounds the nucleus of large lymphocytes. The nucleus of these cells is similar in size and appearance to that of small lymphocytes. The morphological homogeneity of lymphocytes obscures their functional heterogeneity. As is discussed below, lymphocytes participate in multiple aspects of the immune response. Lymphocyte subtypes in blood, see figure 2, are often identified based on their reaction with fluorescent monoclonal antibodies. The majority of circulating lymphocytes are T cells or T lymphocytes, 4 thymus-dependent lymphocytes. These cells participate in certain types of immune responses that do not depend on antibody. T cells comprise 40 to 60% of the total circulating pool of lymphocytes. Subtypes of T cells have been identified using fluorescent monoclonal antibodies to specific cell surface antigens, known as CD antigens. Also possess the common CD3 antigen. So-called helper T cells possess a CD4 antigen cluster, while suppressor T cells lack CD4 but possess CD8. Patients with AIDS show decreased circulating levels of CD4 positive cells. Natural killer NK cells are T lymphocytes that possess the ability to kill tumor cells without prior exposure or priming. Some 20 to 30% of circulating lymphocytes are B cells, which have immunoglobulin or antibody on their surface. B cells are bone marrow-derived lymphocytes. When immunologically activated, they transform into plasma cells that secrete immunoglobulin. Lymphocytes not characteristic of either T cells or B cells are called null cells. The entire scope of the function of null cells, which comprise only 1 to 5% of circulating lymphocytes, is unknown, but it has been established that null cells are capable of destroying tumor cells and virus-infected cells. While B cells mediate immune responses by releasing antibody, T cells often exert their effects by synthesizing and releasing cytokines, hormone-like proteins that act by binding specific receptors on their target cells. Recent research has led to the discovery of many cytokines, with activities ranging from tumor destruction, a function of tumor necrosis factor, to the promotion of blood cell production. Cytokines that limit viral replication in cells, known as interferons, suppress or potentiate the function of T cells, stimulate macrophages, and activate neutrophils. Cytokines that limit viral replication in cells, known as interferons, suppress or potentiate the function of T cells, stimulate macrophages, and activate neutrophils. In some cases, cytokines, like other hormones, can exert potent effects when supplied exogenously. For example, colony-stimulating factors injected into cancer patients can prevent decreases in the production of leukocytes that result from the administration of chemotherapeutic drugs or radiation therapy. The technology of molecular biology is used to produce cytokines for therapy. In this process, sections of lymphocyte DNA containing the gene that codes for the specified cytokine are isolated and then transfected into a bacterial cell, fungus, or rapidly growing mammalian cell. These cells then produce the cytokine and release it into their culture supernatant, from which it can be purified, concentrated, and sterilized for injection. The biological diversity and potency of the cytokines has opened the door to the development of a variety of new pharmacological agents that have proved useful in the treatment of cancer, immune disorders, and other diseases. Blood cells are born in the bone marrow. Mature cells are transient residents of blood. Erythrocytes survive in the circulation for about 120 days, after which they are broken down and their components recycled, as discussed above. Platelets have an average lifespan of 15 to 45 days in the circulation. Many, if not most, of these cells are consumed as they continuously participate in day-to-day hemostasis. The rate of platelet consumption accelerates rapidly during the repair of bleeding caused by trauma. Leukocytes have a variable lifespan. Some lymphocytes circulate for 1 year or longer after production. Neutrophils, constantly guarding body fluids and tissues against infection, have a 6 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. Circulating half-life of only a few hours. Neutrophils and other blood cells must, therefore, be continuously replenished. As mentioned earlier, the process of blood cell generation, hematopoiesis, occurs in healthy adults only in the bone marrow. Extramedullary hematopoiesis, e.g., the generation of blood cells in the spleen, is observed only in some disease states, such as leukemia. Hematopoietic cells are found in high levels in the liver, spleen, and blood of the developing fetus. Shortly before birth, blood cell production gradually begins to shift to the marrow. In newborns, the hematopoietic cell content of the circulating blood is relatively high. Hematopoietic cells are also found in the blood of adults, but in newborns, cells are also found in the blood of adults, but in extremely low numbers. Large numbers of hematopoietic cells can be recovered from aspirates of the iliac crest, sternum, pelvic bones, long bones, and ribs of adults. Within the bones, hematopoietic cells germinate in extravascular sinuses, called neurostroma. Circulating factors and factors released from capillary endothelial cells, stromal fibroblasts, and mature blood cells regulate the generation of immature blood cells from hematopoietic cells and the subsequent differentiation of newly formed immature cells. Blood cell production begins with the proliferation of pluripotent uncommitted stem cells, depending on the stimulating factors. The progeny of pluripotent stem cells may be other uncommitted stem cells or stem cells committed to development along a certain lineage. The committed stem cells include myeloblasts, which form cells of the myeloid series, neutrophils, basophils, and eosinophils, erythroblasts, lymphoblasts, and monoblasts, promoted by hematopoietins and other cytokines. Each of these blast cells differentiates further, a process that ultimately results in the formation of mature blood cells. This is a dynamic process. The hematopoietic cells of the bone marrow are among the most actively reproducing cells of the body. Interruption of hematopoiesis, e.g., by cancer treatment, results in the eventual disappearance of granulocytes from the blood, a condition known as granulocytopenia, or, when specific to neutrophils, neutropenia, in a matter of hours. Platelets disappear next, thrombocytopenia, followed by erythrocytes, a sequence that reflects the circulating lifespan of each cell. Often, hematopoiesis can be restored after its interruption by an infusion of viable hematopoietic cells, e.g., a bone marrow transplant. Administration of committed stem cells shows promise in treating specific blood cell defects hemostasis. Circulating in a high pressure, closed system that communicates with all tissues and cells in the body, blood exchanges oxygen, nutrients, and wastes and provides necessary components for host defense. This communication takes place largely in the complex and dynamic networks of capillary beds that provide oxygen to almost every cell in the body, only the cornea and intervertebral discs are avascular. These tissues receive oxygen by diffusion. Disruption of the integrity of the fragile capillaries may result from minor tissue injury associated with normal physical activity or from massive tissue trauma as a result of serious injury or infection, and may quickly lead to death. Any opening in the vascular network may lead to massive bruising or blood loss if left unrepaired. To minimize bleeding and prevent blood loss after tissue injury, components of the hemostatic system are activated. The components of this dynamic, integrated system include blood platelets, endothelial cells, and plasma coagulation factors. They may be activated on exposure to foreign surfaces during bleeding, or by torn tissue at the site of injury, or by products released from the interior of damaged cells. Hemostasis can be viewed as four separate but interrelated events. Equals compression and vasoconstriction, which act immediately to stop the flow of blood equals formation of a platelet plug. Equals blood coagulation. Equals clot retraction. Physical factors immediately act to constrain bleeding. Immediately after tissue injury, blood flow through the disrupted vessel is slowed by the interplay of several important physical factors, including compression or back pressure exerted by the tissue around the injured area, and vasoconstriction. The degree of compression varies in different tissues. For example, bleeding below the eye is not readily deterred because the skin in this area is easily distensible. Back pressure increases as blood which leaks out of the disrupted capillaries accumulates. In some tissues, notably the uterus after childbirth, contraction of underlying muscles compresses blood vessels supplying the tissue and minimizes blood loss. Damaged cells at the site of tissue injury release potent substances that directly cause blood vessels to constrict, including serotonin, thromboxane E2, epinephrine, and fibrinopeptide B. Platelets form a hemostatic plug. Platelets regulate bleeding in three stages. First, they form multicellular aggregates linked by protein strands at sites of openings in blood vessels. The aggregates form a physical barrier that begins to limit blood loss soon after the opening occurs. Second, phospholipids on the platelet plasma membrane activate the enzyme thrombin, which initiates a cascade of events ending in clot formation. Finally, platelets possess multiple storage granules, which they discharge secrete to enhance coagulation. Platelet activation results in the sequential responses of adherence, aggregation, and secretion. Adherence is initiated when one or more substances, released from cells are activated in plasma at the site of a hemorrhage, bind to receptors in the platelet plasma membrane. Receptor binding results via second messengers in adherence to other platelets in the inner endothelial surface of blood vessels and secretion. Disruption of the endothelium at sites of tissue injury exposes a variety of proteins in the subendothelial matrix, such as collagen and laminin, which either induce or support platelet adherence. Endothelial cells also rapidly deploy cellular adherence antigens known as integrins on the outer surface of their plasma membranes during wound healing. These adherence antigens are deployed to the cell membrane by cellular processes set in motion by factors generated during coagulation or by factors. Institute for Advanced Studies, Jodhpur 7. Unit 7 System Physiology Animal. Released from platelets during clotting. In turn, activated endothelial cells release substances that participate in hemostasis. Von Willebrand factor, a protein synthesized by endothelial cells and megakaryocytes, enhances platelet adherence by forming a bridge between cell surface receptors and collagen in the subendothelial matrix. The protein thrombin, which is generated by the plasma coagulation cascade, is a potent activator of platelet adherence and secretion. Ruptured cells at the site of tissue injury release adenosine diphosphate (ADP), which causes platelets to aggregate at the damaged site. These aggregates effectively stop the flow of blood from the ruptured vessels. Blood coagulation results in the production of fibrin. Platelet aggregates are trapped in a highly organized, firm, and degradable network of fibrin and insoluble protein generated in plasma as a consequence of activation of either the intrinsic or extrinsic clotting cascades discussed below. The fibrin network traps red cells, leukocytes, platelets, and serum at sites of vascular damage, thereby forming a blood clot. The stable, fibrin-based blood clot eventually replaces the unstable platelet aggregate formed immediately after tissue injury. Fibrin is an insoluble polymer of proteolytic products of the plasma protein fibrinogen. Fibrin molecules are cleaved from fibrinogen by thrombin, which is generated in plasma during clotting. In the initial step of fibrin formation, thrombin cleaves four small peptides, fibrinopeptides, from each molecule of fibrinogen. The fibrinogen molecule devoid of these fibrinopeptides is called fibrin monomer. The fibrin monomer spontaneously assemble into ordered fibers arrays of fibrin, resulting in an insoluble matrix of fibrous strands. At this stage, the clot bonds held together by non-covalent forces. A plasma enzyme, fibrin stabilizing factor, factor 13, catalyzes the formation of covalent bonds between strands of polymerized fibrin, stabilizing and tightening the blood clot. The coagulation cascade. Blood clotting is mediated by the sequential activation of a series of coagulation factors, proteins synthesized in the liver that circulate in the plasma in an active state. They are referred to by number designated by Roman numeral in a sequence based on the order of the discovery of each factor. The plasma coagulation factors and their common names are listed in table 3. The sequential activation of a series of inactive molecules resulting in a biological response is called a metabolic cascade. The sequential activation of coagulation factors resulting in the conversion of fibrinogen to fibrin and, hence, clotting is called the coagulation cascade. The deficiency or deletion of any one factor of the cascade has severe consequences. Individuals deficient in factor VIII antihemophilic factor, for example, display prolonged bleeding time on tissue injury as a result of delayed clotting. Those who lack factor VIII have hemophilia, a condition resulting in severe coagulation defects. Two separate coagulation cascades result in blood clotting in different circumstances. The two systems are the intrinsic coagulation pathway and the extrinsic coagulation pathway, figure 4. The final steps in fibrin formation are common to both pathways. In the intrinsic pathway, all the factors required for coagulation are present in the circulation. For initiation of the extrinsic pathway, a factor extrinsic to blood but released from injured tissue, called tissue thromboplastin or tissue factor, factor 3, is required. Phospholipids are required for activation of both coagulation pathways. Phospholipids provide a surface for the efficient interaction of several factors. A component of tissue factor provides the necessary phospholipid for the extrinsic pathway. Phospholipids required for the activation of the intrinsic pathway are found on platelet membranes. Table factors of the coagulation cascade. Scientific name, common name, other names. Factor fibrinogen. Factor I1 prothrombin. Factor 111 tissue thromboplastin tilde tissue factor. Factor B calcium. Factor B proaxellin labile factor, factor 7 proconvertin serum prothrombin. Conversion accelerator SPCA. Factor 8 antihemophilic factor, platelet cofactor 1 factor X Christmas factor, platelet thromboplastin. Component. Factor X Stewart factor. Factor X1 plasma thromboplastin antecedent. Factor 12 Hagman factor, contact factor, factor 13 fibrin stabilizing factor. The final events leading to fibrin formation by both pathways result from the activation of the common pathway. The common pathway is initiated by the conversion of an active clotting factor X to its active form, factor X at C figure 4, and results in the conversion of prothrombin to thrombin, thereby catalyzing the generation of fibrin. Thrombin also enhances the activity of clotting factors B and 8, accelerating upstream events in the coagulation pathway. Finally, thrombin is a potent platelet and endothelial cell stimulus and enhances the participation of these cells in coagulation. Factor X is activated during both the extrinsic and the intrinsic pathways. In the extrinsic pathway, factor X is activated by a complex consisting of activated factor 7, Ca, and factor 3 tissue factor. Activation of this complex by tissue factor bypass is a requirement for coagulation. Factors 8, X, 11, and 12 used in the intrinsic pathway. In the intrinsic pathway, clotting is initiated by the activation of factor 12 by contact to exposed surfaces, such as collagen in the subendothelial matrix. The activation of factor 12 requires several cofactors, including kallikrein and high molecular weight kininogen. In this pathway, factor X is activated by a complex consisting of factor 8, factor IXA, platelet factor 3, and Ca. Any attempt to describe a distinct division of coagulation into two separate pathways is an oversimplification, and the cascade theory has been extensively modified. 8 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. Intrinsic extrinsic pathway. ZIL, XLLA. X greater than XX1A. X3 Cas plus PIXR ovula less than, VIL. Ca2+. Phil. Common pathway prothrombin. *R thrombin to factor. X Euro X. XL. XLLLA. Fibrinogen. Fibrin. Stable fibrin polymer. STCPs in the coagulation cascade. The extrinsic pathway is initiated by tissue factor 1I released from damaged cells. In the presence of CA trademark, factor L converts factor 7 to factor DLLA, which then forms a complex with factor I1L and CA. This complex converts factor X to factor XA. In the intrinsic system, factor XLI is first converted to factor XLLA following its exposure to foreign surfaces, such as the endothelial matrix. Factor XLLA initiates a cascade of events, including activation of factor X, subsequent conversion of thrombin to thrombin, and, finally, fibrin formation. There are many points of interaction between the two pathways, and no one pathway will account for hemostasis. For example, thrombin generated during activation of the extrinsic pathway is an essential cofactor for factor VIII of the intrinsic pathway. Factor DLLA of the extrinsic pathway directly activates factor X of the intrinsic system. Factor 7 can be activated by factors IXA, XA, and XLLA and thrombin. The many additional points of interaction are beyond the scope of this discussion, but the concept of independently acting intrinsic versus extrinsic coagulation pathways has been abandoned. However, the activity of the intrinsic system and the extrinsic system are monitored individually in clinical coagulation tests for diagnostic purposes. The test used to monitor activity of the intrinsic system is the partial thromboplastin time (PTT). The extrinsic system is evaluated by determination of the prothrombin time (PT). In the liver, spleen, and blood of the developing fetus. Shortly before birth, blood cell production gradually begins to shift to the marrow. In newborns, the hematopoietic cell content of the circulating blood is relatively high. Hematopoietic cells are also found in the blood of adults, but in extremely low numbers. Large numbers of hematopoietic cells can be recovered from aspirates of the iliac crest, sternum, pelvic bones, long bones, and ribs of adults. Within the bones, hematopoietic cells germinate in extravascular sinuses, called marrostroma. Circulating factors and factors released from capillary endothelial cells, stromal fibroblasts, and mature blood cells regulate the generation of immature blood cells from hematopoietic cells and the subsequent differentiation of newly formed immature cells. Blood cell production begins with the proliferation of pluripotent uncommitted stem cells, depending on the stimulating factors. The progeny of pluripotent stem cells may be other uncommitted stem cells or stem cells committed to development along a certain lineage. The committed stem cells include myeloblasts, which form cells of the myeloid series, neutrophils, basophils, and eosinophils, erythroblasts, lymphoblasts, and monoblasts, promoted by hematopoietins and other cytokines. Each of these blast cells differentiates further, a process that ultimately results in the formation of mature blood cells. This is a dynamic process. The hematopoietic cells of the bone marrow are among the most actively reproducing cells of the body. Interruption of hematopoiesis, e.g., by cancer treatment, results in the eventual disappearance of granulocytes from the blood, a condition known as granulocytopenia, or, when specific to neutrophils, neutropenia, in a matter of hours. Platelets disappear next, thrombocytopenia, followed by erythrocytes, a sequence that reflects the circulating lifespan of each cell. Often, hematopoiesis can be restored after its interruption by an infusion of viable hematopoietic cells, e.g., a bone marrow transplant. Administration of committed stem cells shows promise in treating specific blood cell defects hemostasis. Circulating in a high pressure, closed system that communicates with all tissues and cells in the body, blood exchanges oxygen, nutrients, and wastes and provides necessary components for host defense. This communication takes place largely in the complex and dynamic networks of capillary beds that provide oxygen to almost every cell in the body. Only the cornea and intervertebral discs are avascular. These tissues receive oxygen by diffusion. Disruption of the integrity of the fragile capillaries may result from minor tissue injury associated with normal physical activity or from massive tissue trauma as a result of serious injury or infection, and may quickly lead to death. Any opening in the vascular network may lead to massive bruising or blood loss if left unrepaired. To minimize bleeding and prevent blood loss after tissue injury, components of the hemostatic system are activated. The components of this dynamic, integrated system include blood platelets, endothelial cells, and plasma coagulation factors. They may be activated on exposure to foreign surfaces during bleeding, or by torn tissue at the site of injury, or by products released from the interior of damaged cells. Hemostasis can be viewed as four separate but interrelated events. Equals compression and vasoconstriction, which act immediately to stop the flow of blood, equals formation of a platelet plug, equals blood coagulation, equals clot retraction. Physical factors immediately act to constrain bleeding. Immediately after tissue injury, blood flow through the disrupted vessel is slowed by the interplay of several important physical factors, including compression or back pressure exerted by the tissue around the injured area, and vasoconstriction. The degree of compression varies in different tissues. For example, bleeding below the eye is not readily deterred because the skin in this area is easily distensible. Back pressure increases as blood which leaks out of the disrupted capillaries accumulates. In some tissues, notably the uterus after childbirth, contraction of underlying muscles compresses blood vessels supplying the tissue and minimizes blood loss. Damaged cells at the site of damaged cells at the site of tissue injury release potent substances that directly cause blood vessels to constrict, including serotonin, thromboxane E2, epinephrine, and fibrinopeptide B. Platelets form a hemostatic plug. Platelets regulate bleeding in three stages. First, they form multicellular aggregates linked by protein strands at sites of openings in blood vessels. The aggregates form a physical barrier that begins to limit blood loss soon after the opening occurs. Second, phospholipids on the platelet plasma membrane activate the enzyme thrombin, which initiates a cascade of events ending in clot formation. Finally, platelets possess multiple storage granules, which they discharge secrete to enhance coagulation. Platelet activation results in the sequential responses of adherence, aggregation, and secretion. Adherence is initiated when one or more substances, released from cells are activated in plasma at the site of a hemorrhage, bind to receptors in the platelet plasma membrane. Receptor binding results via second messengers in adherence to other platelets in the inner endothelial surface of blood vessels and secretion. Disruption of the endothelium at sites of tissue injury exposes a variety of proteins in the subendothelial matrix, such as collagen and laminin, which either induce or support platelet adherence. Endothelial cells also rapidly deploy cellular adherence antigens known as integrins on the outer surface of their plasma membranes during wound healing. These adherence antigens are deployed to the cell membrane by cellular processes set in motion by factors generated during coagulation or by factors. Institute for Advanced Studies, Jodhpur 7. Unit 7 System Physiology Animal. Released from platelets during clotting. In turn, activated endothelial cells release substances that participate in hemostasis. Von Willebrand factor, a protein synthesized by endothelial cells and megakaryocytes, enhances platelet adherence by forming a bridge between cell surface receptors and collagen in the subendothelial matrix. The protein thrombin, which is generated by the plasma coagulation cascade, is a potent activator of platelet adherence and secretion. Ruptured cells at the site of tissue injury release adenosine diphosphate (ADP), which causes platelets to aggregate at the damaged site. These aggregates effectively stop the flow of blood from the ruptured vessels. Blood coagulation results in the production of fibrin. Platelet aggregates are trapped in a highly organized, firm, and degradable network of fibrin and insoluble protein generated in plasma as a consequence of activation of either the intrinsic or extrinsic clotting cascades discussed below. The fibrin network traps red cells, leukocytes, platelets, and serum at sites of vascular damage, thereby forming a blood clot. The stable, fibrin-based blood clot eventually replaces the unstable platelet aggregate formed immediately after tissue injury. Fibrin is an insoluble polymer of proteolytic products of the plasma protein fibrinogen. Fibrin molecules are cleaved from fibrinogen by thrombin, which is generated in plasma during clotting. In the initial step of fibrin formation, thrombin cleaves 4 small peptides, fibrinopeptides, from each molecule of fibrinogen. The fibrinogen molecule devoid of these fibrinopeptides is called fibrin monomer. The fibrin monomers spontaneously assemble into ordered fibrous arrays of fibrin, resulting in an insoluble matrix of fibrous strands. At this stage, the clade I bonds held together by non-covalent forces. A plasma enzyme, fibrin stabilizing factor, factor 13, catalyzes the formation of covalent bonds between strands of polymerized fibrin, stabilizing and tightening the blood clot. The coagulation cascade. Blood clotting is mediated by the sequential activation of a series of coagulation factors, proteins synthesized in the liver that circulate in the plasma in an active state. They are referred to by number designated by Roman numeral in a sequence based on the order of the discovery of each factor. The plasma coagulation factors and their common names are listed in table 3. The sequential activation of a series of inactive molecules resulting in a biological response is called the metabolic cascade. The sequential activation of coagulation factors resulting in the conversion of fibrinogen to fibrin and, hence, clotting is called the coagulation cascade. The deficiency or deletion of any one factor of the cascade has severe consequences. Individuals deficient in factor VIII antihemophilic factor, for example, display prolonged bleeding time on tissue injury as a result of delayed clotting. Those who lack factor VIII have hemophilia, a condition resulting in severe coagulation defects. Two separate coagulation cascades result in blood clotting in different circumstances. The two systems are the intrinsic coagulation pathway and the extrinsic coagulation pathway, figure 4. The final steps in fibrin formation are common to both pathways. In the intrinsic pathway, all the factors required for coagulation are present in the circulation. For initiation of the extrinsic pathway, a factor extrinsic to blood but released from injured tissue, called tissue thromboplastin or tissue factor, factor 3, is required. Phospholipids are required for activation of both coagulation pathways. Phospholipids provide a surface for the efficient interaction of several factors. A component of tissue factor provides the necessary phospholipid for the extrinsic pathway. Phospholipids required for the activation of the intrinsic pathway are found on platelet membranes. Table factors of the coagulation cascade. Scientific name, common name, other names. Factor fibrinogen. Factor I1 prothrombin. Factor 111 tissue thromboplastin tilde tissue factor. Factor B calcium. Factor B proaxellin labile factor 7 proconvertin serum prothrombin. Conversion accelerator SPCA. Factor 8 antihemophilic factor platelet cofactor 1 factor X Christmas factor platelet thromboplastin. Component. Factor X Stewart factor. Factor X1 plasma thromboplastin antecedent. Factor 12 Hagman factor contact factor factor 13 fibrin stabilizing factor. The final events leading to fibrin formation by both pathways result from the activation of the common pathway. The common pathway is initiated by the conversion of an active clotting factor X to its active form, factor X of C figure 4, and results in the conversion of prothrombin to thrombin, thereby catalyzing the generation of fibrin. Thrombin also enhances the activity of clotting factors B and 8, accelerating upstream events in the coagulation pathway. Finally, thrombin is a potent platelet and endothelial cell stimulus and enhances the participation of these cells in coagulation. Factor X is activated during both the extrinsic and the intrinsic pathways. In the extrinsic pathway, factor X is activated by a complex consisting of activated factor 7, Ca, and factor 3 tissue factor. Activation of this complex by tissue factor bypass is the requirement for coagulation. Factors 8, X, 11, and 12 used in the intrinsic pathway. In the intrinsic pathway, clotting is initiated by the activation of factor 12 by contact to exposed surfaces, such as collagen in the subendothelial matrix. The activation of factor 12 requires several cofactors, including kallikrein and high molecular weight kininogen. In this pathway, factor X is activated by a complex consisting of factor 8, factor IXA, platelet factor 3, and Ca. Any attempt to describe a distinct division of coagulation into two separate pathways is an oversimplification, and the cascade theory has been extensively modified. 8 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. Intrinsic extrinsic pathway. ZIL, XLLA. X, greater than XX1A. X3 Cas plus PIXR ovula less than, VIL. CA2 plus. VIL. Common pathway prothrombin, asterisk R thrombin to factor. X Euro, X. XL. XLLA. Fibrinogen, fibrin. Stable fibrin polymer. STCPs in the coagulation cascade. The extrinsic pathway is initiated by tissue factor, factor 1I released from damaged cells. In the presence of CA trademark, factor IL converts factor 7 to factor VLLA, which then forms a complex with factor I1L and CA. This complex converts factor X to factor XA. In the intrinsic system, factor XLI is first converted to factor XLLA following its exposure to foreign surfaces, such as the subendothelial matrix. Factor XLLA initiates a cascade of events, including activation of factor X, subsequent conversion of thrombin to thrombin, and, finally, fibrin formation. There are many points of interaction between the two pathways, and no one pathway will account for hemostasis. For example, thrombin generated during activation of the extrinsic pathway is an essential cofactor for factor 8 of the intrinsic pathway. Factor VLLA of the extrinsic pathway directly activates factor X of the intrinsic system. Factor 7 can be activated by factors IXA, EXA, and XLLA and thrombin. The many additional points of interaction are beyond the scope of this discussion, but the concept of independently acting intrinsic versus extrinsic coagulation pathways has been abandoned. However, the activity of the intrinsic system and the extrinsic system are monitored individually in clinical coagulation tests for diagnostic purposes. The test used to monitor activity of the intrinsic system is the partial thromboplastin time (PTT). The extrinsic system is evaluated by determination of the prothrombin time (PT). To a large extent, the interaction of coagulation factors occurs on the surfaces of platelets and endothelial cells. While plasma can eventually clot in the absence of surface contact, localization and assembly of coagulation factors on cell surfaces amplifies reaction rates by several orders of magnitude. Clot retraction is a phenomenon that usually occurs within minutes or hours after clot formation. The clot draws together, extruding a very large fraction of the serum. The retraction requires platelets. Clot retraction decreases the breakdown of the clot and enhances wound healing. Institute for Advanced Studies, Jodhpur 9. Unit 7 System Physiology Animal. B. Cardiovascular system. Comparative anatomy of heart structure. Myogenic heart. Specialized tissue. ECG its principal insignificance. Cardiac cycle. Heart as a pump. Blood pressure. Neural and chemical regulation of all above. Circulatory systems. Pumps, vessels, and blood. A circulatory system consists of a muscular pump, heart, of fluid blood that can transport materials, and a series of conduits, blood vessels through which the fluid can be pumped around the body. Heart, blood, and vessels are also known collectively as a cardiovascular system, from the Greek cardia, heart, and the Latin vasculum, small vessel. In this section, we will compare the circulatory systems of different groups of animals. Some simple aquatic animals do not have circulatory systems. A circulatory system is unnecessary if the cells of an organism are close enough to the external environment that nutrients, respiratory gases, and waste can diffuse between the cells and the environment. Small aquatic invertebrates have structures and body shapes that permit direct exchanges between cells and environment. Many of these animals have flattened body shapes that maximize the amount of surface area that is in contact with the external environment. See figure 1a. In arthropods, tissue fluid re-enters the heart through the ostia. 1. Insect if. Tubular heart. B. Mollusk. Stomach artery to foot. Vein from foot. In molluscs, a system of vessels drains the intercellular spaces and regions of blood to the heart. S. 1. Open circulatory systems in both arthropods and mole. Mollusks B. Blood is pumped by a tubular heart and directed to differ end regions of the body through vessels that open into intercellular spaces. The cells of some other aquatic invertebrates are served by highly branched central cavities called gastrovascular systems that exchange their contents with the external environment. All the cells of a sponge, for example, are in contact with, or very close to, the water that surrounds the animal and circulates through its central cavity. See figure 1b. Large surface areas and branched internal cavities cannot satisfy the needs of larger animals with many layers of cells. The cells of such animals are surrounded by an Internal environment of extracellular fluids, which we will refer to in this chapter as tissue fluids. Circulatory systems carry materials to and from all regions of the body to maintain the optimum composition of the tissue. Fluids, which in turn serve the needs of the cells. Circulatory systems may or may not keep the circulating fluid blood separate from the tissue fluid. Such circulatory systems are considered closed or open, respectively. Open circulatory systems move tissue fluid. The simplest circulatory system squeeze tissue fluid through intercellular spaces as the animal moves. In these open circulatory systems, there is no distinction between tissue fluid and blood. Usually a heart assists the distribution of the fluid. The contractions of the heart propel the tissue fluid through vessels leading to different regions of the body, but the fluid leaves those vessels to trickle through the tissues and eventually return to the heart. Open circulatory systems are found in arthropods and mollusks as well as in some other invertebrate groups. In the generalized arthropod shown in figure 1a, the fluid returns to the heart through valved openings called ostia. In the mollusk in figure 1b, open vessels aid in the return of tissue fluid to the heart. Closed circulatory systems circulate blood through tissues. In a closed circulatory system, a system of vessels keeps circulating blood separate from the tissue fluid. Blood is pumped through this vascular system by one or more muscular hearts, and some components of the blood never leave the vessels. Closed circulatory systems characterize vertebrates, annelids, and some other invertebrate groups. A simple example of a closed circulatory system is that of the common earthworm, figure 0.2. One large blood vessel on the ventral side of the earthworm carries blood from its anterior end to its posterior end. Smaller vessels branch off and transport the blood to even smaller vessels serving the tissues in each segment of the worm's body. In the smallest vessels, respiratory gases, nutrients, and metabolic waste diffuse between the blood and the tissue fluid. The blood then flows from these vessels into larger vessels that lead into one large vessel on the dorsal side of the worm. The dorsal vessel carries the blood from the posterior to the anterior end of the body. 5 pairs of vessels connect the large dorsal and ventral vessels in the anterior end, thus completing the circuit. The dorsal vessel and the 5 connecting vessels serve as hearts for the earthworm. Their contractions keep the 10 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. Blood circulating. The direction of circulation 1 is determined by one-way valves in the dorsal and connecting vessels. Connecting vessels, ventral vessel. To a closed circulatory system, an earthworm exemplifies a closed circulatory system in which blood is confined to the blood vessels, is kept separate from tissue fluids, and is pumped by one or more muscular hearts. Closed circulatory systems have several advantages over open systems. The blood can flow more rapidly through vessels and through intercellular spaces, and can therefore transport nutrients and waste to and from tissues more rapidly. By changing resistance in the vessels, closed systems can be more selective in directing blood to specific tissues. These specialized cells and large molecules that aid in the transport of hormones and nutrients can be kept within the vessels, but can drop their cargo in the tissues where it is needed. Overall, closed circulatory systems can support higher levels of metabolic activity than open systems can, especially in larger animals. How, then, do highly active insect species achieve high levels of metabolic output with their open circulatory systems? One way is by not depending on their circulatory systems for respiratory gas exchange. Vertebrate circulatory systems. Vertebrates have closed circulatory systems and hearts with 2 or more chambers. Valves between the chambers and between the chambers and the vessels prevent the backflow of blood when the heart contracts. As we explore the features of the circulatory systems of the different classes of vertebrates, a general evolutionary theme will become apparent. There is a progressively more complete separation of the blood that circulates to the gas exchange organs from the blood that circulates to the rest of the body. In fish, blood is pumped from the heart to the gills and then to the tissues of the body and back to the heart. In birds and mammals, at the other end of the evolutionary scale, blood is pumped from the heart to the lungs and back to the heart in a pulmonary circuit, and then from the heart to the rest of the body and back to the heart in a systemic circuit. We will trace the evolution of the separation of the circulation into 2 circuits. The closed vascular system of vertebrates begins with vessels called arteries that carry blood away from the heart. Arteries give rise to smaller vessels called arterioles, which feed blood into capillary beds. Capillaries are the tiny, thin-walled vessels where materials are exchanged between the blood and the tissue fluid. Small vessels called venules drain capillary beds. The venules join together to form larger vessels called veins, which deliver blood back to the heart. We can trace the evolutionary history of vertebrate circulatory systems by examining the circulatory systems of fish, lungfishes, amphibians, reptiles, crocodilians, and mammals. Fish have 2-chambered hearts. The fish heart has 2 chambers. An atrium receives blood from the body and pumps it into a more muscular chamber, the ventricle. The ventricle pumps the blood to the gills, where gases are exchanged. Blood leaving the gills collects in a large dorsal artery, the aorta, which distributes blood to smaller arteries and arterioles leading to all the organs and tissues of the body. In the tissues, blood flows through beds of tiny capillaries, collects in venules and veins, and eventually returns to the atrium of the heart. Oxygenated blood. Deoxygenated blood. Fish have a heart with 2 chambers, a single atrium and a single ventricle. Most of the pressure imparted to the blood by the contraction of the ventricle is dissipated by the high resistance of the narrow spaces through which blood flows in the gill lamellae. As a result, blood leaving the gills and entering the aorta is under low pressure, limiting the maximum capacity of the fish circulatory system to supply the tissues with oxygen and nutrients. This limitation on arterial blood pressure does not seem to hamper the performance of many rapidly swimming species, such as tuna and marlin. However, the evolutionary transition from breathing water to breathing air had important consequences for the vertebrate circulatory system. An example of how the system changed to serve a primitive lung can be seen in the African lungfish. Lungfish are periodically exposed to water with low oxygen content or to situations in which their aquatic environment dries up. Their adaptation for dealing with these conditions is an outpocketing of the gut that serves as a lung. The lung contains many thin-walled blood vessels, so blood flowing through those vessels can pick up oxygen from air gulped into the lung. Institute for Advanced Studies, Jodhpur 11. Unit 7 System Physiology Animal. Deoxygenated blood I, L deoxygenated II. Blood ventricle, Li, L mixed blood atria. The lungfish heart has 2 atria, 1 receiving oxygenated blood from the lung and 1 deoxygenated blood from the body. How does the circulatory system take advantage of this new organ? The posterior pair of gill arteries has been modified to carry blood to the lung, and a new vessel carries oxygenated blood from the lung back to the heart. In addition, 2 anterior gill arches have lost their gills, and their blood vessels deliver blood from the heart directly to the dorsal aorta. Because a few of the gill arches retain gills, the African lungfish can breathe either air or water. The lungfish heart has adaptations that partially separate the flow of its blood into pulmonary and systemic circuits. Unlike other fishes, the lungfish has a partly divided atrium. The left side receives oxygenated blood from the lungs, and the right side receives deoxygenated blood from the other tissues. These 2 blood streams stay mostly separate as they flow through the ventricle and a large vessel leading to the gill arches. As a result, oxygenated blood mostly goes to the anterior gill arteries leading to the dorsal aorta, and the deoxygenated blood mostly goes to the posterior arches with functional gills into the lung. We can conclude that the lung of the lungfish evolved as a means of supplementing oxygen uptake from the gills, but the associated modifications of the vascular system set the stage for the evolution of separate pulmonary and systemic circulations. Amphibians have 3-chambered hearts. Pulmonary and systemic circulation are partly separated in adult amphibians. A single ventricle pumps blood to the lungs and to the rest of the body. 2 atria receive blood returning to the heart. 1 receives oxygenated blood from the lungs, and the other receives deoxygenated blood from the body. Deoxygenated blood I, L deoxygenated blood L mixed blood. In adult amphibians, the pulmonary Y and D systemic circuits are partially separated. The heart has 3 chambers. Reptiles have exquisite control of pulmonary and systemic circulation. Turtles, snakes, and lizards are commonly said to have 3-chambered hearts, while crocodilians, crocodiles, and alligators are said to have 4-chambered hearts. But this statement is an oversimplification. The hearts of all these animals have 2 separate atria and a ventricle that is divided in a complex way so that mixing of oxygenated and deoxygenated blood is minimized. I, originated loon. Blood in right aorta. I, deoxygenated blood left atrium. Mixed blue aorta. The reptilian ventricle is partly divided by a septum, which directs oxygenated blood to the body and deoxygenated blood to the lungs. The most important and unusual feature of reptilian and crocodilian hearts is their ability to alter the proportion of blood going to the lungs and to the rest of the body. Consider the behavior, ecology, and physiology of these animals. Despite the common image of turtles as being slow and plodding, reptiles and crocodilians can be fast, active, powerful animals. They can also be inactive for long periods of time, during which they have metabolic rates much lower than the resting metabolic rates of birds and mammals. The enormous range of metabolic needs in these animals means that they do not have to breathe continuously. Some species are also accomplished divers and spend long periods underwater, where they cannot breathe. To understand the adaptations of the reptilian and crocodilian hearts, you have to realize that there is no benefit in sending blood to the lungs when an animal is not breathing. The hearts of these animals circulate blood through their lungs and then to the rest of their bodies when they are breathing, but when they are not breathing, they can bypass the pulmonary circuit and pump all the blood around the body. How do they accomplish this switching? Reptiles have two aortas instead of one. The right aorta can receive blood from either the right side or the left side of the ventricle. The two sides of the ventricle are partially divided by a septum. When the animal is breathing air, two factors cause blood from the right side of the ventricle to go preferentially into the pulmonary circuit rather than into the systemic circuit. First, the resistance in the pulmonary circuit is lower than that in the systemic circuit. Second, there is a slight asynchrony in the timing of ventricular contraction, so the blood in the right side of the ventricle tends to be ejected slightly before the blood in the left side. As the ventricle contracts, the deoxygenated blood in the right side of the ventricle moves first into the pulmonary circuit. When the oxygenated blood in the left side of the ventricle starts to move, it encounters resistance in the pulmonary circuit, which is already filled with the deoxygenated blood from the right side. Therefore, the blood from the left side tends to flow into the two aortas. When the reptile stops breathing, blood flow is rerouted by constriction of vessels in the lung. As resistance in the pulmonary circuit increases, the blood from the right side of the ventricle tends to be directed into one of the aortas. 12 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. As a result, blood from both sides of the ventricle flows through the aortas to the systemic circuit. The ability of snakes, lizards, and turtles to redirect blood flow from the pulmonary circuit to the systemic circuit depends on the incomplete division of their ventricles. Crocodilians have true four-chambered hearts with completely divided ventricles. Yet the crocodilians have not lost the ability to shunt blood from the pulmonary circuit when they are not breathing. The crocodilians have one aorta originating in the left ventricle and one aorta originating in the right ventricle. However, a short channel connects these two aortas just after they leave the heart, making it possible for blood to flow from one aorta to the other. Because the crocodilians' ventricles are separate, they can generate different pressures when they contract. When the animal is breathing, the pressure in the left ventricle and the left aorta is higher than the pressure in the right ventricle. This higher pressure is communicated through the connecting channel to the right aorta and prevents right ventricle blood from entering that aorta. As a result, both aortas carry blood from the left ventricle, and the blood from the right ventricle flows to the pulmonary circuit. Deoxygenated lung. Blood right eye deoxygenated aorta. Blood. Mixed blood tilde left aorta. Crocodilians have completely divided ventricles, but they still have the ability to alter the distribution of blood to the pulmonary and systemic circuits because of the connection between two of the major vessels leaving the heart. When a crocodilian is not breathing, constriction of vessels in the lung increases the resistance in the pulmonary circuit. As a result, pressure builds up in the right ventricle to a level that exceeds the pressure in the right aorta. Under these conditions, blood from both ventricles flows through the two aortas in the systemic circuit, and little blood flows into the pulmonary circuit. Birds and mammals have fully separated pulmonary and systemic circuits. The four-chambered hearts of birds and mammals completely separate their pulmonary and systemic circuits. Separate circuits have several advantages. Deoxygenated and deoxygenated blood cannot mix. Therefore, the systemic circuit is always receiving blood with the highest oxygen content. The respiratory gas exchange ones maximized because the blood with the lowest oxygen content and highest CO2 content is sent to the lungs. These separate systemic and pulmonary circuits can operate at different pressures. Lung. I. L oxygenated. Bleed W0 pulmonary I. Deoxygenated R. Circuit blood equals P1 L left. Birds and mammals underscore. FJ ventricle have four chambered equals D hearts. Their pulmonary tilde right. And systemic circuits four ventricle. Are totally separate. Systemic circuit. The tissues of birds and mammals have high nutrient demands and thus a very high density of the smallest vessels, the capillaries. Many small vessels present high resistance to the flow of blood. Therefore, high pressure is required in the systemic circuits of birds and mammals. Their pulmonary circuits have fewer capillaries and thus lower resistance than their systemic circuits, so the pulmonary circuits of birds and mammals can function at lower pressures. The human heart, two pumps in one. Like all other mammalian hearts, the human heart has four chambers, two atria and two ventricles, figure 3. The atrium and ventricle on the right side of your body are called the right heart. The atrium and ventricle on the left side of your body are called the left heart. The right heart pumps blood through the pulmonary circuit, and the left heart pumps blood through the systemic circuit. Valves D between the atria and ventricles, the atrioventricular valves, prevent backflow of blood into the atria when the ventricles contract. The pulmonary valve and the aortic valve, positioned between the ventricles and the major arteries, prevent the backflow of blood into the ventricles. In this section, we'll first focus on the flow of blood through the heart and through the body. Then we'll examine the unique electrical properties of cardiac muscle that result in the heartbeat, and we'll see how the heart's electrical activity can be recorded in an ECG electrocardiogram. Blood flows from right heart to lungs to left heart to body. Let's follow the circulation of the blood through the heart, starting in the right heart. The right atrium receives deoxygenated blood from the superior upper vena cava and the inferior lower vena cava, see figure 3, large veins that collect blood from the upper and lower body, respectively. The veins of the heart itself also drain into the right atrium. From the right atrium, the blood flows into the right ventricle. Most of the filling of the ventricle results from passive flow while the heart is relaxed between beats. Just at the end of this period of ventricular filling, the atrium contracts and adds a little more blood to the ventricular volume. The right ventricle then contracts, pumping the blood into the pulmonary artery, which transports it to the lungs. After gas exchange occurs in the lungs, the pulmonary veins return the oxygenated blood from the lungs to the left atrium, from which the blood enters the left ventricle. As with the right side of the heart, most left ventricular filling is passive, but the ventricle is topped off by Institute for Advanced Studies, Jodhpur 13. Unit 7 System Physiology Animal. Contraction of the atrium just at the end of the period of passive filling. The walls of the left ventricle are powerful muscles that contract around the blood with a ringing motion starting from the bottom. When pressure in the left ventricle is high enough to push open the aortic valve, the blood rushes into the aorta to begin its circulation throughout the body. In figure 3, observe vessels shown inward being oxygenated IKL around the lungs to the left heart. We punch at 0 OT rear EV80. WH. That the left ventricle is more massive than the right ventricle. The left ventricle has to propel the blood through many more kilometers of blood vessels into the right ventricle and must therefore push against more resistance, even though both pump the same volume of blood. 3. The human heart and circulation in the human heart. Blocked fields from right fear to lungs 1 O left heart 1 O body. The atrioventricular valves prevent blood from passing by the ITEX. The IVC ventricles contract. CT pulmonary AMD sortic valves prevent blood from flowing back into ventricles from the arterius WH to event and right S relax. Pulmonary valve. Superacer. WLR. From less than 4 lung. Digitogenated blood from the KS of the body. Appers the right atrium. Dot dot. Il telos strogan aovercula val RBA I ventricle. TH and Ott Vantrison. Pumping Resthea blood into L. The pulmonary sent integral Schwaring Blueberg. Decogenated bio from the body. I.e. ready heart. Which pump N1. D the lungs for oxygenation. Both sides of the heart contract at the same time. The contraction of the two atria, followed by the contraction of the two ventricles and then relaxation, is called the cardiac cycle. Contraction of the ventricles is called ventricular systole, and relaxation of the ventricles called ventricular diastole. Figure 4. Just at the end of diastole, the atria contract and top off the volume of blood in the ventricles. The sounds of the cardiac cycle, the lub dub, heard through a stethoscope placed on the chest, are created by the slamming shut of the heart valves. The closing and opening of these valves are simple mechanical events resulting from pressure differences on the two sides of the valves. As the ventricles begin to contract, the pressure in the ventricles rises above the pressure in the atria. Blood starts flowing back into the atria, and the atrioventricular valves close. Lub. When the ventricles begin to relax, the high pressure in the aorta and pulmonary artery causes blood to start to flow back into the ventricles, and this flow of blood closes the aortic and pulmonary valves. Dub. Defective valves produce turbulent blood flow and produce the sounds known as heart murmurs. 4. Example, if an atrioventricular valve wants defective blood. E. Synculk, E. Biud recerns. I for mu, PFL, M. 1 O the Beft atrium. Atrioventricular valve. FL flows through an NTVBT ventricle. Will flow back into the atrium with a whoosh sound. Following the lub. The cardiac cycle can be felt in the pulsation of arteries such as the one that supplies blood to your hand. You can feel your pulse by placing 2 fingers from one hand lightly over the wrist of the other hand just below the thumb. During systole, blood surges through the arteries of your arm and hand, and you can feel the surge of the pulsing of the artery in your wrist. Blood pressure changes associated with the cardiac cycle can be measured in a large artery in your arm by using an inflatable pressure cuff and a pressure gauge, together called a sphygmomanometer, and a stethoscope figure 5. This method measures the minimum pressure necessary to compress an artery so that blood does not flow through it at all, the systolic value, and the minimum pressure that permits intermittent flow through the artery, the diastolic value. In a conventional blood pressure reading, the systolic value is placed over the diastolic value. Normal values for a young adult might be 120 mm of mercury Hg during systole and 80 mm Hg during diastole, or 120 80ths. 14 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. Count, dub, the ventricles relax, lub, the ventricles proturine, the ventricles contract th, the atrioventricular falls at the end of systole. Valves close, and pressure in and since pressure Js now blood is pumped OUWT. The ventricles builds up until the ventrix AMD greater in the aorta, the. At the after contract, the aortic and pulmonary into the aorta and aortic and pulmonary vibes VT ventricles fill. Underscore, valves open, pulmonary artery, slam SHUL with blood, BI. Colon dot, LI. Left atrium. Right atrium approximately 5. Left ventricle. Diastole. 130 milliliters. Pressure in left ventricle, volume in and left. At B. 2 Olumla W ventricle, ML. Closing parenthesis. Pressure in aorta, mmHg. Opening curly bracket, closing parenthesis. 65 milliliters. UO10 0.30405 10.7 L. 0910 times seconds. For the cardiac cycle, the rhythmic contraction systole and relaxation diastole of the ventricles is called the cardiac cycle. The graphical representation below shows pressure and volume changes during the cardiac cycle for the left ventricle only. Potential stimulates contraction. Large groups of cardiac muscle cells contract in unison. This coordinated contraction is essential for pumping blood effectively. Second, some cardiac muscle cells are pacemaker cells. These cells have the ability to initiate action potentials without stimulation from the nervous system. When they fire action potentials, they stimulate neighboring cells to contract. The primary pacemaker of the heart is a nodule of modified cardiac muscle cells, the sinoatrial node, located at the junction of the superior vena cava and right atrium. The resting membrane potentials of these cells are not stable, but gradually becomes less negative, more positive until they reach the threshold for initiating an action potential. Figure 6. Prosu in RTE colas increase and presiers in SCUITIS arsizli laura presiris rover the sound of Biud becomes continuous. At this time the cuff TS. Just below the distal pressure in the artery. This Parsons blood pressure is 120,170. To dose both. Whelms. No sound is audible. THTRECO on TG on H in the RTCKRAY road. At this time, press ISNU the RC is just below. The peak systole. Press RTAA artery. 5 measuring blood pressure in the major the action potentials of pacemaker cells result from the artery of the arm can be measured with a device called a sphygmo penning of voltage gated calcium channels, so they look. Manometer, which combines an inflatable cuff and a pressure gauge, different from the sodium action potentials. They are. A stethoscope is also used to detect sounds created by the blood slower and last longer. The nervous system controls the vessels in response to changes in pressure during the cardiac cycle. Heartbeat speeds it up or slows it down by influencing the rate at which pacemaker cells gradually depolarize between action potentials. Acetylcholine released by parasympathetic neurons onto the pacemaker cells slows the rate of depolarization and thereby slows the heart rate. Norepinephrine released by sympathetic neurons onto the pacemaker cells increases the rate of depolarization and thereby speeds the heart rate. See figure 6. The heartbeat originates in the cardiac muscle. Cardiac muscle has some unique properties that allow it to function as an effective. Cardiac muscle has some unique properties that allow it to function as an effective pump. First, cardiac muscle cells are in electrical contact with one another through gap junctions, which enable action potentials to spread rapidly from cell to cell. Because of spreading action. Institute for Advanced Studies, Jodhpur 15. Unit 7 System Physiology Animal. The action potential initiated in the atria is regenerated. Sympathetic parasympathetic autonomic activity autonomic and conducted to and through the ventricular muscle. Increases the activity, slows the mass by a system of modified, non-contractile cardiac. Heartbeat rate. J. Heartbeat rate. Muscle cells. Situated at the junction of the atria and the plus 61 underscore L ventricles is a nodule of modified cardiac muscle cells. Called the atrioventricular node, which is stimulated by E, LNR, LFI, the depolarization of the atria. E equals heartbeat. 23 rate with a slight delay. It generates action potentials that are 2E conducted to the ventricles via a bundle of fibers called 2 threshold, the bundle of His. These fibers divide into right and left. Bundle branches that run to the tips of the ventricles and equals 70. L then spread throughout the ventricular muscle mass is O248, 1012 per J fibers. These conducting fibers ensure that the time sec cardiac action potential spreads rapidly and evenly throughout the ventricular muscle mass, starting at the very bottom of the ventricles. The short delay in the spread of the action potential imposed by the atrioventricular node ensures that the atria contract before the ventricles do, so that the blood passes. 6. The autonomic nervous system controls heart rate. The plasma membranes of pacemaker cells spontaneously depolarize to threshold and fire action potentials. Signals from the 2 divisions of the autonomic nervous system raise and lower the heart rate by altering the rate at which the cells depolarize. Their effects can be seen in the altered slope of the membrane potential curve prior to reaching threshold. A conduction system coordinates the contraction of heart muscle. A normal heartbeat begins with an action potential in the sinoatrial node, figure 7. This action potential spreads rapidly throughout the electrically coupled cells of the atria, causing them to contract in unison. Since there are no gap junctions between the cells of the atria and those of the ventricles, however, the action potential does not spread directly to the ventricles, and the ventricles do not contract in unison with the atria. Widths. Heart at rest diastole. Progressively from the atria to the ventricles to the arteries. Electrical properties of ventricular muscle sustain heart contraction. Contractions of ventricular muscle fibers last for about 300 milliseconds, much longer than those of skeletal muscle fibers. The reason is found in the electrical properties of these cells. Like neuronal and skeletal muscle action potentials, ventricular muscle cell action potentials are initiated by the opening of voltage-gated sodium channels. Unlike neurons and skeletal muscle fibers, however, ventricular muscle cells remain depolarized for a long time. This plateau of the action potential is due to the sustained opening of voltage-gated calcium channels, figure 8. The contraction of cardiac atria S of his muscle, like other muscle, is stimulated when Ca is S1 and available to bind with troponin. As long as their Ca, Pi bundle channels remain open, the ventricular muscle cells equals branches. Sinoatrial node continue to contract. Pacemaker B M for J. Fibers ventricular muscle cells atrioventricular node ventricles remain depolarized for a plateau period following the firing of an action potential. Atrial contraction, fluorite. I. The sinoatrial node fires, 100, 0 equals X. Action potential spread, underscore F is for the atria, percent equals. Which contract? E, MNA asterisk channels G2. E equals 5 2 version 1. 0 0 equals S0 E2% GC count T equals T2. GE ventricular contraction B section. Systole equals S 3 OPJ 100 equals. The atrioventricular no add RE minus 1. A. I. 0015030 fires. Sending impulses L tilde. Along conducting fibers. The ventricles contract. A time seconds. 8. The action potential of ventricular muscle fibers. The rising phase of the action potential of ventricular muscle fibers graphed in. Red is due to the opening of voltage-gated not channels blue. However, T membrane remains in a depolarized state for a prolonged time because of the opening of voltage-gated CA channels black. 7. The heartbeat pacemaker cells in the sinoatrial node initiate the heartbeat by firing action potentials. 16. Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. The ECG records the electrical activity of the heart. Electrical events in the cardiac muscle during the cardiac cycle can be recorded by electrodes placed on the surface of the body. Such a recording is called an electrocardiogram, ECG. EFR free wing. E. Aneries have lots S. Of Eastishires L8. Tilde, K, basal lamina H, and HI equals. L or EKG, W EKG, Beck. Lose the Gregg 5 equals, a Leslie C Lyle AYER, equals F8 and ELR low I word for heart is cardia. The ECG1 is SL low and less than sinus muscle. Heva Hesta an important tool for diagnosing heart. Third 10 withstand IE Beckido problems. IGH pressures, S. The action potentials that sweep through the muscles of the atria and the ventricles before they contract are such. Massive, localized electrical events that they cause electric currents to flow outward from the heart to all parts of the body. Surface electrodes placed at EY high different locations on the body detect those electric currents at different times and therefore register a voltage difference. The appearance of the ECG depends on the placement of the electrodes. Electrodes placed on the right wrist and left ankle produce the low normal ECG shown in figure 9. The wave patterns of the ECG are designated P, Q, R, S, and T, each letter representing a particular event in the cardiac muscle, as shown in the figure. Large, small arterioles, capillaries, venules, wings. 10. Anatomy of blood vessels. The anatomical characteristics of blood vessels match steeler functions. The walls of the large arteries have many collagen and elastic fibers, which enable them to withstand the high pressures of blood flowing rapidly from the heart. Figure 10. The elastic fibers have another important function as well. During systole, they are stretched and thereby store some of the energy imparted to the blood by the heart. During diastole, they return this energy to the blood by QR and section together correspond to the depolarization of the ventricles. T corresponds to the relaxation and repolarization of the ventricles. P corresponds to the depolarization of the atrial muscle. G underscore elastic recoil, squeezing it and pushing it forward. As AL in result, even though the flow of blood through the arteries 1.0 R pulsates with the beating of the heart, it is smoother than it would be through a system of rigid pipes. Smooth muscle cells in the walls of the arteries and arterioles allow those vessels to be constricted or dilated. O5S5SS when the diameter of the vessels changes, their high resistance to blood flow changes as well, and the amount arterial of blood flowing through them changes as a result. By pressure influencing the contraction and relaxation of the smooth muscle in the vessel walls, neuronal and hormonal mechanisms can control the resistance of the vessels and therefore the distribution of blood to the different tissues of the body. The arteries and arterioles are referred to as FIP 121F5F Fed Love Dub time sec. The sounds heard through a stethoscope GCCURGT the beginning and end of systole resistance vessels because their resistance can vary. 9. Electrocardiogram and EKG can be used to monitor heart blood flow slowly through capillary beds function. Variations from the normal pattern shown here can be used to diagnose heart problems. Beds of capillaries lie between arterioles and venules. No cell of the body is more than a couple of cell diameters away from a capillary. The needs of the cells are served by the exchange of materials between blood and tissue. The vascular system, arteries, capillaries, and veins. Blood circulates throughout the body in a system of blood vessels, arteries, capillaries, and veins. In this section, we will see how the structure of each of these vessel type supports its functions. We will also consider another set of vessels, the lymphatic vessels, which return tissue fluid to the blood. Arteries and arterioles have abundant elastic and muscle fibers. Fluid across the capillary walls. Capillaries have thin, permeable walls, and blood flows through these vessels slowly, facilitating this exchange. Figure 11. To anyone who has played with a garden hose, it may seem strange that blood flows through the large arteries rapidly at high pressures, but when it reaches the small capillaries, the pressure and rate of flow decrease. When Institute for Advanced Studies, Jodhpur 17. Unit 7 System Physiology Animal. You restrict the diameter of a garden hose by placing your thumb over the opening. The pressure in the hose increases, which in turn increases the velocity of the water spraying out of the hose. This puzzle is solved by two more pieces of information. First, arterioles are highly branched. When flow through one branch is restricted, blood flows into other branches, so pressure does not build up quickly. Second, each arteriole gives rise to a large number of capillaries. Even though each capillary has a diameter so small that red blood cells must pass through in single file, there are so many capillaries that their total cross-sectional area is much greater than that of any other class of vessels. As a result, all of the capillaries together have a much greater capacity for blood than do the arterioles. Returning to our garden hose analogy, if we connected the hose to many junctions leading to small irrigation tubes, the pressure and the flow in each of the irrigation tubes would be quite low. 11. Narrow lane capillaries have a very small diameter, and flow flows through them slowly. Materials are exchanged in capillary beds by filtration, osmosis, and diffusion. The walls of capillaries are made of a single layer of thin endothelial cells. In most tissues of the body other than the brain, capillaries have tiny holes called fenestrations, Latin, windows. Capillaries are permeable to water, to some ions, and to some small molecules, but not to large molecules such as proteins. Blood pressure therefore squeezes water and some small solutes out of the capillaries and into the surrounding intercellular spaces. Why don't water and small molecular weight solutes collect in the intercellular spaces? How is the blood volume maintained if fluid is continuously leaking out of the capillaries? An answer to this question was put forth more than 100 years ago by the physiologist E.H. Starling. Starling suggested that water balance in capillary beds is a result of two opposing forces, which have come to be known as Starling's forces. One force is blood pressure, which squeezes water and small solutes out of the capillaries, and the other is osmotic pressure created by the large protein molecules that cannot leave the capillaries. Starling called this second force colloidal osmotic pressure. He hypothesized that blood pressure is high at the arterial end of a capillary bed and drops steadily as blood flows to the venous end. Figure 12. The colloidal osmotic pressure, however, is constant along the capillary. As long as the blood pressure is above the osmotic pressure, water leaves the capillary, but when blood pressure falls below the osmotic pressure, water returns to the capillary. The actual numbers for a normal capillary bed in a resting person suggest that there would be a slight net loss of water to the intercellular spaces. Several observations supported Starling's model. In people with severe liver disease or protein starvation, there is a fall in blood protein concentration that leads to an accumulation of water in the extracellular spaces, which results in tissue swelling or edema. Edema is also a characteristic of the inflammation reaction that accompanies tissue damage or allergic responses. Histamine, a mediator of inflammation that is released by certain white blood cells, increases the permeability of capillaries and also relaxes the smooth muscles of the arterioles, resulting in higher blood pressure in the capillaries. The hypothesis modeled in figure 12 predicts that edema should occur in all of these cases. As fluid is squeezed out of the fluid is pulled back into the capillary L Y ab R low Y odd pressure, capillary by osmotic pressure. Blood colloidal pressure, osmotic 40 pressure, 18 pressure. 25. Venule end. MHG. Blood pressure 40, blood pressure 16, osmotic pressure equals 25, osmotic pressure equals 25, net outward force 15, net inward force minus 9. B50. Net driving force for B fluid to leave capillary. OB blood pressure. YBRX. Equal osmotic net driving force for BP pressure fluid to re-enter capillary. OL. 12 Starling's forces. Starling's model explains how blood volume is maintained in the capillary beds. 8. When blood pressure is greater than the colloidal osmotic pressure, water leaves the capillary. When blood pressure falls below this osmotic pressure, water returns to the capillary. 8. The balance of these two forces changes over the capillary bed as blood pressure falls. Only recently have Starling's forces come into question as a complete explanation for fluid exchange in capillary beds. There are situations that are not explained by Starling's hypothesis. During strenuous exercise, the blood pressure in the arterioles serving the muscles rises substantially, yet edema does not occur. In birds, the blood pressure in arterioles is much higher than in mammals, and the colloidal osmotic pressure is lower. If edema is not a chronic problem in exercising muscles and in birds, what is missing from Starling's model? Recent research suggests that bicarbonate ions, HCO, in the blood plasma are an important contributor to the osmotic attraction of water back into the capillary. As the blood flows through the capillary, Colorado diffuses into the plasma and is converted into bicarbonate ions, therefore. 18 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. There is a substantial rise in HCO concentration as blood flows through the capillary. When a person is at rest, the increasing HCO trademark concentration can cause the osmotic pressure of the blood at the venous end to be 30 mm Hg higher than at the arterial end, and during strenuous exercise this difference can be hundreds of mHg. Thus it appears that Colorado and HCO trademark are the major factors that pull water back into the capillaries, not colloidal osmotic pressure. Lipid-soluble substances and many small solute molecules can easily pass through capillary walls from an area of higher concentration to one of lower concentration. The capillaries in different tissues, however, are differentially selective as to the sizes of molecules that can pass through them. All capillaries are permeable to O, Colorado, glucose, lactate, and small ions such as Na and C. The capillaries of the brain do not have fenestrations, and therefore not much else can pass through them other than lipid-soluble substances, such as alcohol. This high selectivity of brain capillaries is known as the blood-brain barrier. Much less selective capillaries are found in the digestive tract, where nutrients are absorbed, and in the kidneys, where wastes are filtered. Blood flows back to the heart through veins. The pressure of the blood flowing from capillaries to venules is extremely low, and is insufficient to propel blood back to the heart. The walls of veins are more expandable than the walls of arteries, and blood tends to accumulate in veins. As much as 80% of the total blood volume may be in the veins of a resting individual. Because of their high capacity to store blood, veins are called capacitance vessels. Blood flow through veins that are above the level of the heart is assisted by gravity. Below the level of the heart, however, venous return is against gravity. The most important force propelling blood from these regions is the squeezing of the veins by the contractions of surrounding skeletal muscles. As muscles contract, the vessels are compressed, and blood is squeezed through them. Blood flow may be temporarily obstructed during a prolonged muscle contraction, but when muscles relax, blood is free to move again. One-way valves within the veins of the extremities prevent backflow of blood. Thus, whenever a vein is squeezed, blood is propelled forward toward the heart. Figure 13. In a resting person, gravity causes blood accumulation in the veins of the lower body and exerts back pressure on the capillary beds. This back pressure shifts the balance between blood pressure and osmotic pressure so that there is an increased loss of fluid to the intercellular spaces. That is why you have trouble putting your shoes back on after you sit for a long time with your shoes off, such as on an airline flight. In persons with very expandable veins, the veins may become so stretched that the valves can no longer prevent backflow. This condition produces varicose, swollen veins. Draining of these veins is highly desirable and can be aided by wearing support hose and periodically elevating the legs above the level of the heart. When an animal walks or runs, its legs act as auxiliary vascular pumps, returning blood to the heart from the veins of the lower body. As a greater volume of blood is returned to the heart contracts more forcefully, and its pumping action is enhanced. This strengthening of the heart beat I once due to a property of cardiac muscle cells described by the Frank Starling law. If the cells are stretched, as they are when the volume of returning blood increases, they contract more forcefully. Contractions of skeletal muscles squeeze the vein. Muscle contracts. Valve closed hash, valve open. TT. This squeezing moves the blood in the veins toward the heart because of one-way valves that prevent backflow. Muscle relaxes. Valve open, valve closed. End trademark. Blood is propelled forward by back pressures due to contractions, muscle contractions, and, in atria, contractions of muscles, some body regions, by gravity. And, in some regions, by gravity. A. 13 one-way flow veins have valves that prevent blood from flowing backward. The actions of breathing also help return venous blood to the heart. The ventilatory muscles create negative pressure that pulls air into the lungs. See figure 11, and this negative pressure also pulls blood toward the chest, increasing venous return to the right atrium. In addition, some of the largest veins closest to the heart contain smooth muscle that contracts at the onset of exercise. This contraction can rapidly increase venous return and stimulate the heart in accord with the Frank Starling law, thus increasing cardiac output. Lymphatic vessels return tissue fluid to the blood. The tissue fluid that accumulates outside the capillaries contains water and small molecules, but no red blood cells, and less protein than there is in blood. A separate system of vessels, the lymphatic system, returns tissue fluid to the blood. After entering the lymphatic vessels, the tissue fluid is called lymph. Fine lymphatic capillaries merge progressively into larger and larger vessels and end in two lymphatic vessels, the thoracic ducts, that empty into large veins at the base of the neck. The left thoracic duct carries most of the lymph from the lower part of the body and is much larger than the right thoracic duct. Lymph, like blood, is propelled toward the heart by skeletal muscle contractions and breathing movements, and lymphatic vessels, like veins, have one-way valves that keep the lymph flowing toward the thoracic duct. Institute for Advanced Studies, Jodhpur 19. Unit 7 System Physiology Animal. Mammals and birds have lymph nodes along the major lymphatic vessels. Lymph nodes are a major site of lymphocyte production and of the phagocytic action that removes microorganisms and other foreign materials from the circulation. The lymph nodes also act as filters. Particles become trapped there and are digested by phagocytes in the nodes. When you get an infection, the lymph nodes closest to the infection become swollen and sore. The swelling is due to the accumulation of immune system cells that have been activated to fight the infection. Will you die of cardiovascular disease? Cardiovascular disease is responsible for about half of all deaths each year in the United States and Europe. It is by far the largest single killer in those countries. The immediate cause of most of these deaths is heart attack or stroke, but those events are frequently the end result of a disease called atherosclerosis, hardening of the arteries, that begins many years before symptoms are detected. Hence atherosclerosis is called a silent killer. Healthy arteries have a smooth internal lining of endothelial cells, figure 14a. Smooth muscle endothelium. 14 atherosclerotic plaque, A healthy, clear artery. B an atherosclerotic artery, clogged with plaque and a thrombus. This lining can be damaged by chronic high blood pressure, smoking. B an atherosclerotic artery, clogged with plaque and a thrombus. This lining can be damaged by chronic high blood pressure, smoking, a high-fat diet, or microorganisms. Deposits called plaque begin to form at sites of endothelial damage. First, the damaged endothelial cells attract certain white blood cells to the site. These cells are then joined by smooth muscle cells migrating from the deeper layers of the arterial wall. Lipids, especially cholesterol, are deposited in these cells, so that the developing plaque becomes fatty, fibrous connective. Tissue made by the invading smooth muscle cells in the plaque, along with deposits of calcium, makes the artery wall less elastic, hence, hardening of the arteries. The growing plaque deposit narrows the artery and causes turbulence in the blood flowing over it. Local conditions can cause blood platelets to stick to the plaque and initiate the formation of an intravascular blood clot. called a thrombus, which can quickly block the artery. Figure 14b. The blood supply to the heart itself flows through the coronary arteries, which are highly susceptible to atherosclerosis. As these arteries narrow, blood flow to the heart muscles decreases. Chest pain and shortness of breath during mild exertion are symptoms of this condition. A person with atherosclerosis is at high risk of forming a thrombus in a coronary artery. This condition, called coronary thrombosis, can totally block the vessel, causing a heart attack or myocardial infarction. MI. A piece of a thrombus that breaks loose, called an embolus, is likely to travel to and become lodged in a vessel of smaller diameter, blocking its flow and embolism.